Hello everyone! Every cell in our body contains an extraordinary amount of genetic information. For many years, accessing this information was a slow, expensive, and technically demanding process that allowed scientists to sequence only relatively small portions of DNA. So, have you ever wondered which technology has revolutionized DNA sequencing? Welcome to the course Bioinformatics and Genomic Data Analysis. Today, we'll start our first lesson on next-generation sequencing, commonly referred to as NGS. Nowadays, DNA sequencing is at the heart of modern biology and medicine. It helps scientists identify disease-causing mutations, study infectious pathogens, understand genetic diversity, and develop personalized treatments. However, this was not always possible. Until recently, sequencing DNA was slow, expensive, and limited to relatively small DNA fragments. Through our rich table of contents, we will discover how NGS revolutionized DNA sequencing. Before discussing NGS, let's briefly review the technology that came before it. For many years, Sanger sequencing was considered the gold standard for DNA sequencing. Sanger technology determines the DNA sequence using what we call chain-terminating nucleotides. And this technique is known for its excellent accuracy, because it relies on the selective incorporation of chain-terminating dideoxynucleotides during DNA synthesis. Despite its reliability and its crucial role in many landmark discoveries, Sanger sequencing has important limitations. Since it processes only one DNA fragment at a time, making it relatively slow, labor-intensive, and expensive for large-scale projects. These limitations motivated the development of faster and more efficient sequencing technologies. In contrast to Sanger technology, NGS completely transformed its paradigm. Let's see how it emerged. Next-generation sequencing introduced a completely different concept known as massively parallel sequencing. Advances in capillary electrophoresis, fluorescence detection, and automated data analysis contributed to the development of these new high-throughput technologies. So, instead of sequencing one DNA fragment after another, NGS analyzes millions of fragments simultaneously during a single run. This innovation dramatically increases sequencing speed while reducing the cost and generating enormous amounts of genomic data. Today, NGS has become the standard platform for large-scale genomic studies as well as transcriptomics. Now, let's see together the principles of NGS technologies. Although different NGS platforms use different sequencing technologies, they all share the same fundamental principle. Next-generation sequencing, also known as high-throughput sequencing, generates millions of short DNA sequencing in a single sequencing run. These sequences are called reads. Typically, Illumina produces reads of about 125 base pairs. Each read represents only a small fragment of a genome or a transcriptome. These short reads are then computationally assembled and analyzed using bioinformatics tools to reconstruct the original genetic information. Now, regarding NGS approaches, NGS can be applied in three different ways depending on the biological question. Whole genome sequencing analyzes the complete genome and provides the most comprehensive information. Whole exome sequencing focuses on protein coding regions where many disease-causing mutations are found. And finally, targeted sequencing examines selected genes or genomic regions, making it faster and more cost-effective for many clinical applications. Choosing the appropriate approach always depends on the objectives of your study. Now, an important question arises. What can we actually do with all the sequencing data from NGS? Take a moment to reflect. And now, let's dive together into the key analysis enabled by NGS. NGS enables a wide variety of genomic analysis. For example, we can reconstruct entire genomes or transcripts through de novo assembly, even when no reference genome is available. We can also detect genetic variations, such as single nucleotide variants, insertions, or deletions, which are essential for studying genetic diseases and evolution. In addition, NGS allows us to analyze genome architecture, including copy number variations and gene expression using RNA sequencing. Finally, comparative genomics, which enables researchers to compare genomes from different species or individuals, helping them understand evolution, biodiversity, and gene function. We will explore many of these analyses in greater detail throughout the course. Let's now walk through the general NGS workflow. Regardless of the sequencing platform, the process can be summarized into 4 key steps. The first stage involves DNA fragmentation, where genomic DNA is broken into small molecules suitable for sequencing. The second stage is library preparation, during which sequencing adapters are attached to each fragment, making them compatible with the sequencing platform. The library is then loaded into the sequencing instrument. The third stage is the sequencing process itself. During this step, the instrument determines the nucleotide sequence of millions of DNA fragments simultaneously. Finally, the sequence output undergoes bioinformatics analysis. The generated reads are processed, aligned to reference genome, compared and interpreted to exact biologically relevant information. In other terms, bioinformatics transform the raw sequencing data into meaningful biological information through quality control, alignment, variant detection, and data interpretation. The success of NGS is largely explained by its wide range of applications, especially in genetic diseases diagnostics, personalized medicine and drug development, and finally, evolutionary and population studies. In clinical genetics, NGS enables the identification of disease-causing mutations and supports the diagnostics of rare genetic disorders. In oncology, NGS helps characterize tumors and guides personalized treatment strategies. To conclude, DNA sequencing has evolved from Sanger to NGS and then to third-generation technologies, making genomic analysis faster, more accurate, and more accessible while advancing research, diagnostics, and precision medicine. We reached the end of this lesson, so thank you for your attention, and I look forward to seeing you in the next one. I wish you an enjoyable learning experience and every success in your studies.